ready?
Yes, sir.
Okay, let's go.
See yourself on here. Let me see yourself on. Oh, okay. Oh, okay. I got you. Well, as you approach, and then you just get out there. And I think the mic will pick you up. All right, everyone may be seated. I'm sorry. Ladies and gentlemen, this witness has been previously sworn to testify truthfully. Mr. Goble.
Thank you, Your Honor. Dr. Bigler, could you state your name for the record, please?
Yes, my name is Erin, E-R-I-N, David Bigler, B as in boy, I-G-L-E-R.
And you don't have to go into detail. So the court's given you permission to testify remotely for some health issues, right?
That is correct.
What are your major areas of training and expertise?
Well, I'm Professor Emeritus of Psychology and Neuroscience at Brigham Young University, and I was formerly Professor of Psychology and Neuroscience at the University of Texas at Austin. Also held an appointment at the University of Texas Health Science Center in San Antonio when I was in Texas. I'm a clinical neuropsychologist, licensed, board certified in that specialty, and I'm a cognitive neuroscientist and researcher as well as a clinician.
And does that include expertise in neuropsychology and neurophysiology and neurophysiological diagnostics and neuroimaging?
Yes, those are all of my specialty areas. I was a postdoctoral fellow at the Barrow Neurological Institute in Phoenix, Arizona before taking my position at the University of Texas and the State Hospital there in Austin. And then I was recruited back to Brigham Young University to help develop their neuroscience program and all of those specialty areas that you just mentioned.
And have you been published in those fields?
Yes, I've been a professor all those years and directed, I established and directed the brain imaging and behavior laboratory. I also developed the magnetic resonance imaging research center at Brigham Young University and directed that until my retirement in 2018 as a professor and director of large laboratories involved in research. I've published extensively in the peer-reviewed literature on the interface of brain imaging and behavior, which was the name of my laboratory.
Did you also publish textbooks or book chapters?
Yes, I've published 12 different textbooks, working on the 6th edition of one of the textbooks that I've co-authored, and 80 some odd book chapters and over 400 peer-reviewed articles in the field.
Have you received any awards or scholarships?
Yes, I've received a number of awards, both nationally and internationally, and I've lectured nationally and internationally and lectured at most of the major universities here in the United States.
And have you received any grants or fellowships?
Yes. As I mentioned, I fellowship trained in neurophysiology and neuropsychology. That was a long time ago, 1974 to 77. and I've been a co-investigator on numerous grants over the years. They're detailed in my vita.
And did you examine Tanner Horn?
I did not.
So were you asked to give any specific opinions or diagnoses of Tanner?
I was not.
Were you asked to testify regarding brain anatomy and brain functioning in neuropsychiatric disorders generally?
Yes, that was what I was asked to do to assist his counsel in understanding brain anatomy and brain function and contemporary methods for evaluating structure and function of the human brain.
Okay, and did you review some materials in preparation for testifying?
I did.
And did you observe Dr. Lewine's testimony earlier today?
I did.
So it was kind of a mouthful for what we asked you to do. We brought up to the jury in our opening about how important the brain is. Did we ask you really to educate on how the brain and behavior interrelate?
Yes. In fact, the motto of my research lab was every behavior has an anatomy. The behavior emerges first in the brain and then outwardly in behavior.
Now, I have a model brain here, so you can direct me if you want to use that to explain anything. But can you please start by describing brain anatomy and brain function for the jury?
Yeah, so I believe that the model that you have is, I have an older version of the model that you have, and I'm holding that up here. We saw some diagrams in Dr. Lewine's presentation. This is a model. So it typically starts with the 4 lobes. So this is the frontal lobe, this is the parietal lobe, this is the occipital lobe, and I'm holding the brain here by the temporal lobe, and down here is the cerebellum, and this is the brainstem. And then if we turn it, this is called medial, if we turn it medially here, now we're looking at the inner part of the left hemisphere. And there's a couple of important areas here that were talked about. This area here is that orbital frontal area that Dr. Lewine was talking about. And then this area right here that looks like a curve here, or a bridge. In fact, that's actually the Latin part of it. It's the fornix. This is the fornix. And the back of the fornix comes out of the temporal lobe, and then it dives into a structure here called the thalamus, and then there's projection that comes up into the cortex, but in particular to this area right here, which is called the cingulus gyrus here. So that's a brief overview of some of the main areas of the brain, and I'm happy to discuss their function in more detail if need be.
Yeah, can you talk briefly? I think Dr. Lewine mentioned but didn't go into much detail about how different parts of the brain, he gave a few examples, different parts of the brain control different functions, and he mentioned sort of networks. Can you talk some about what neural networks are?
Yes, neural networks are very important because the brain functions always as a unit. Dr. Lewine mentioned that the brain processes 100 trillion signals every moment of our existence. It's incredibly complex. There's nothing more complex than the human brain. And so you have these major hubs in the brain. And then you have these nodes that connect to the hubs. Dr. Lewine used those particular terms. He also showed telephone poles or power lines. Now, of course, we have cell phones, so it's a little bit of an older analogy, but the connector part of the telephone analogy, that is that axon, and he showed a diagram of that axon. In the classroom, I would demonstrate it like this. So I'd hold my arm up. This is the cell body. This is where, of the neuron, this is where all the metabolic machinery is taking place. These are the dendrites. They're communicating with other axons. This is where the synapse occurs. And then there's one singular process that comes out of the cell body. It can branch into multiple areas, but then it terminates in the synaptic area to communicate with the next. And so you have major hubs and you have minor hubs, and they form these networks because they connect to all of these different nodes. So there's not just one area dedicated in the brain that carries out function. It's multiple areas working contiguously and simultaneously to generate first perceptions and initial processing, and then to actually generate behavior. I always like to use the analogy of roads, highways, freeways, interstates. When I was a professor at the University of Texas, I would drive up to Dallas often. I did things with the different universities up there. And, I-35 is a nice direct route until you get just outside of Dallas. And then all these other highways, freeways, and interstates start to connect. So that's a hub. Fort Worth is a big hub. But you can go anywhere in the United States now once you get to one of those hubs. The brain is exactly the same way. Everything is interconnected.
So can you describe, I guess, kind of how that, I understand the analogy, but how does that end up controlling behavior through the way that those networks connect?
Well, each one of those hubs, each one of those particular networks is subserving a different aspect of behavior. And so one of the areas that Dr. Lewine spoke of is this orbital frontal area. The reason it's called the orbital frontal area is it sits just above the orbit, the eye socket. And so this was referred to as the orbital frontal area because this is the frontal part of the brain. Well, this orbital frontal area has a direct connection with the temporal lobe. And the temporal lobe has these brain structures called the limbic system. And they're regulators of emotion, including emotional tone. This area up here is called the dorsal, lateral, and then as you move forward, prefrontal cortex. This is the slower, more contemplative, decision-making part of the brain. This is the more emotional, reactive part of the brain. If there's an imbalance between these areas, then emotion can override contemplative behavior. And this is one of the things that Dr. Lewine was talking about in terms of social cognition, because social cognition gets tied in to emotional processing, emotional regulation, and emotional tone.
Now, I can throw in a little question the state had earlier. Are there sometimes, or some ways that the brain can try to overcome any sort of issues with the networks?
Yes, Well, again, some of the things from, well, let me start back with the brain develops as an experience-dependent organ. So it interacts with the environment, and the environment changes the brain interacts with the environment. It's a back and forth process that takes place. This upper part of the frontal cortex, which is slower and contemplative, and the temporal area and the orbital frontal area are emotionally reactive. So one of the old adages of counting to 10, for example, before you say something, is allowing the frontal lobe to take better control and dampen down emotions. But if emotions arise, then they cut off that contemplative part of the frontal cortex, and inappropriate behaviors come from that.
So when you have the disciplines of psychology, psychiatry, medicine, trying to deal with psychiatric disorders or developmental disorders, is that where they're actually affecting the brain, correct?
Yes, that's where, all behavioral function, and from the standpoint of psychiatric disorders, it's really brain health. Psychiatric disorders are not some ethereal thing that's happening independent of the brain. They are, in fact, related to the brain. Excuse me.
And I believe Dr. Lewine referred to the philosophical idea of the mind, but we're talking about the physical brain and how it directly affects how we behave.
That's correct, because the brain is involved in perception. You have the perception, and then Dr. Lewine used the example of language processing, that in the right-handed individual typically starts here in the temporal area, but for it to actually be expressed as language, that has to communicate up here. Using various imaging techniques, one in the laboratory can actually watch that process happen, and then the behavior emerges, but it's the brain that's driving that behavior.
Tell the jury more about that. Is imaging one of the ways that you detect these, you determine the neural networks? Is that what the research has shown?
Yes, that's a major part of contemporary research looking at cognition and behavior.
And what kind of imaging are we talking about?
All of the imaging that was discussed today, electroencephalography, magnetic resonance imaging, positron emission tomography.
So those are commonly used in scientific and forensic setting and clinical settings.
That's all correct.
So can you please explain how quantitative abnormalities would affect the neural networks?
So where one has to start, if you go back to the actual imaging, Dr. Lewine spoke of the T1 weighted MRI. Well, that MRI is showing you the basic anatomy. In fact, the anatomy is so good on a T1 weighted MRI that it approximates what you would do at postmortem. So you could have a postmortem view and a side-by-side MRI, and you would see that they're very, very similar. But the resolution on MRI just takes you down to a millimeter. Well, in a single cubic millimeter of cortical brain tissue, you have somewhere around 100,000 neurons, and you have somewhere around 5 million synapses just in that little 1 millimeter area. And so when you're looking at the clinical scan, which was discussed this morning, you're looking at the gross anatomy, but you're not looking at the cellular level. You're having to make some inferences about that. And where the quantitative imaging assists is it's like artificial intelligence in a way. It's taking a deeper dive into the data. And as Dr. Lewine explained, that T1 weighted image gives you 3 tissue types. Gray matter, that's where the cell bodies and the synapses occur. White matter, that's that singular process axon that then connects with other cells. And cerebral spinal fluid, CSF. The computer programs allow you to then what's called segment into white matter, gray matter, and cerebral spinal fluid. And then through a variety of very eloquent algorithms, it allows you to then classify those areas. You saw the images that had the colors. Those are classified images that are identifying specific areas of the brain. And now, because this was what's called a volume acquisition MRI, you know the parameters, and you can define these areas, we call them regions of interest. You can define those areas, and then you can quantify their volume. You can actually do even more. You can measure their shape, their contour, the surface area. So there's a number of metrics. The only ones that were applied here were volumetric analyses. But it's giving you a window into a more detailed aspect of brain structure when you're looking at the MRI. And the PET imaging is telling you something more about metabolic function, and the EEG is telling you something more about the physiologic function of the brain. This is what's called multimodality imaging, where you take this imaging and you integrate it to best understand what the imaging data is telling you about that individual.
Okay, and just to clarify real quick, when you're talking about the MRI was equivalent to a postmortem, basically saying if you took a brain out and looked at it and looked at the MRI, those are going to be matched up, right?
That's correct.
And you're talking about the PET scan being metabolic. Can you tell me what that means?
Well, as Dr. Lewine testified, there's a radio tracer that is labeled to glucose. The neuron, and like Dr. Lewine indicated, there's close to 100 billion neurons. And there's another set of cells called glial cells that are just as frequent, sometimes more. So you have in excess of 200 billion cells. They have no capacity to store oxygen or glucose. They always have to have oxygen and glucose. And so if you have a radio tracer that labels the glucose, then you can monitor the uptake. And then as Dr. Lewine was showing, you're from a quantitative PET analysis standpoint, you're looking at either increased or decreased activity from whatever the basic baseline would be in comparison to that neurotypical normative sample.
No, the other thing I was asking, at a more basic level, your brain has to have glucose to function, correct?
Oh, yes.
And that's one of the reasons why testing where that's going and how it's moving is important.
It's measuring the uptake of that radioactive particle as it's being metabolized.
Can you explain to the jury how neural network pathology relates to psychiatric disorders?
Well, one of the very interesting aspects of psychiatric disorders is it almost doesn't matter which psychiatric disorder one is looking at. Some of the key areas of the brain that are typically going to be involved, and that's true of all of the psychiatric disorders that have been discussed concerning Mr. Horner. It involves that inferior frontal area, the temporal lobe, and structures inside the temporal lobe called the hippocampus and the amygdala. The fornix takes that information from the hippocampus and some from the amygdala and transfers it to other parts of the brain. These are parts that are referred to as the limbic system. And so, again, whether we're looking at depression, anxiety disorder, schizophrenia, whatever psychiatric condition we're looking at, the more common aberrations that are shown with these kinds of quantitative and multimodality techniques is that these areas of the brain are affected in some way, at some level.
Now, you mentioned before that Dr. Lewine's combination of those three tests is kind of a model for how you look at neural networks. Is that typical in the forensic and scientific community?
That's correct.
And I believe, let me ask you another question. As far as things that can affect the brain, affect the neural networks, does that include things like toxic exposure, examples of fetal alcohol, lead, or toxic stress?
That's correct.
Things like neurodevelopmental disorders like autism spectrum disorder, ADHD, are those things that you can also show a neurological link?
Correct.
We haven't talked much about psychiatric disorders, but are things like, I guess Lewine may refer to mood disorders, things like that, those are also things that actually are grounded in your brain and your neural network. Is that correct?
Yes, I mean, within those systems that I was talking about, there's a valence, a balance. And if there's underlying neurologic dysfunction, that balance can go off keel. And one can be overly emotional or emotionally reactive or display deficits in judgment or conforming behavior to societal standards. All of those things can happen.
One more thing to clarify, I didn't remember the term, I think it was multimodality. Can you just break that down for me real quick?
Well, yes. So MRI is considered to be structural when you're using that T1 and some of the other imaging sequences. But one of the other things that Dr. Lewine did is the diffusion tensor imaging, which allows you to look at pathways, and you can actually apply what are called diffusion metrics to assess the integrity of those pathways. So that's telling you structure. The PET scan is telling you about metabolic activity, and the EEG is telling you about the physiology of the brain, how the brain is reacting and connecting. And one of the things he talked about, Dr. Lewine talked about, is coherence. So areas that are showing connectivity will have somewhat similar coherence. So it allows you to look at how the network is connecting. And then as you go across these different modalities of imaging, you can make more inferences about how the brain is functioning and how that structural aspect of the brain relates to function.
Now, can you tell the jury a little bit about the difference between the clinical use of those imaging to detect gross abnormalities versus the clinical looking at more the quantitative, those other issues, and the forensic and scientific use of those different imaging?
Well, one of the things that's happening is a movement towards what's called precision medicine, and that's taking advantage of artificial intelligence, the power of these methods for rapidly examining the brain. And this is likely to become standard fare within, in my opinion, in the next 5 to 10 years, so that it's one thing to look at a brain and the first line of assessment of a brain is to see, is there a medical necessity there? Is there a tumor? Is there a vascular lesion? Is there something that needs attention, an infection? Once that is confirmed not to be there, then you're looking at, is there any kind of major developmental abnormality, any other kind of deviation? And when those things are not present, there is a tremendous amount of information that is in that scan, but it's not looked at. But it can be now with the technologies that we have. And so what's happening in cognitive neuroscience, in clinical neuropsychology, is that it's moving in that direction. So, for example, Dr. Lewine mentioned Alzheimer's disease. This is one of the areas where it's really happening quite quickly. So as a neuropsychologist, I can administer tests that can measure memory, but I'm not necessarily looking specifically at the memory network. But with imaging, using especially diffusion tensor imaging, structural quantitative imaging, I can look at the different components of that. And so if I'm working up an Alzheimer's patient, then I want to know that, I don't want to know just that the brain kind of looks normal. I want to know what's the size of the hippocampus, what's the connection of the hippocampus with the rest of the brain, and all of those things. So that's happening right now. It will move into the clinical world, in my opinion, in the next 5 to 10 years. Right now, it's primarily a research tool.
So that's when we're talking about the computer analysis. Is that becoming more available? Is that what you're saying?
Oh, yes. Yeah, I mean, the supercomputers are, so one of the, when I lecture on this, one of the slides I have is that every quantitative aspect of imaging analysis had to be done by hand, tracing the structures. When I started this when I was at the University of Texas. Now, we can do all of these major quantitative analyses in a matter of a few minutes. It would take days to do that on an individual patient or client in the past, and now it can be done in a few minutes. And with supercomputers, it can be even done faster.
So the analysis itself is not new. That's something that's backed by research and studies.
Yes, we first published on quantitative image analysis back in the 1980s.
Here, I'll pass it.
Thank you, Judge. Hi, Dr. Bailey. Bigler, can you hear me?
Yes, I can hear you just fine.
Okay, good. You did not do an evaluation on Tanner Horner, the defendant in this case, correct?
No, I did not see him, evaluate him, or anything else.
Have you ever met Tanner Horner?
No.
So you're here today just to testify, I guess, in general or educational reasons?
Correct.
And you're not testifying that Tanner Horner didn't know right from wrong, are you?
I am not.
And you're not saying that autism caused this crime? Doctor?
I don't know, we heard you.
Did you hear the question?
Oh, right from wrong?
No, I'm sorry. The question was, you're not saying that autism was the cause of the crime in this case.
No.
And we asked this question to all of our experts. But were you paid for your testimony today?
I was.
And what is your total bill in this case?
It's about $4,000.
And is that including your testimony today?
No. It'd be another $1,000 or so for today.
Thank you. I appreciate it.
Read.
Nothing
No,
further.
Judge, thank you.
May, Dr. be excused finally?
Yes, Judge.
Doctor, thank you very much.
You're welcome. Thank you.
Goodbye.